morning, everyone. We're going to go ahead and get started. I'm Sangeeta Bhatia. I'm one of the members of the Koch Institute and excited to be moving into the new building in November. I'm going to be your session moderator this morning. The session is on detecting and modeling cancer. So to get started, I'm going to be introducing Joe DeSimone. He's our first speaker. He's a professor of chemistry. He received his PhD in chemistry from Virginia Polytech under the guidance of James McGrath. He joined the Faculty of Chemistry at University of North Carolina, Chapel Hill in 1990, where he's been ever since. He's currently the Chancellor's Eminent Professor of Chemistry and the founding director of the Institute for Nanomedicine there. His work spans many areas of nanomedicine, most recently the systematic study of the impact of nanoparticle shape on biological interactions and many other areas. He's the co-founder of Liquidia Technologies, which uses a templating method to manufacture engineered nanoparticles for life sciences and material applications. Among his many, many awards are the Lemelson MIT Prize, awarded here, given for inventors dedicated to improving our world through technological invention and innovation, the NIH Director's Pioneer Award. He's also a fellow of the American Academy of Arts and Sciences. He's a friend to the Koch Institute, as he's sat on our advisory board since last year, so we'll be happily seeing much more of him in the years to come. The title of his talk today is Nano-fabrication Technologies for the Production of Vaccines and Therapeutics. Joe, welcome.
Good morning, everyone. Thank you for inviting me. It's terrific to be back and see a lot of great friends, and want to thank Tyler and Bob for the invitation back. My relationship with Bob has been for some time. This is a fully bioabsorbable stent that we developed with a company about 8 years ago, and it was great to work closely with him. That's in about 1,100 people today in an international clinical trial, so it's been a great relationship to continue working with Bob. But this morning I want to tell you about a story related to nanoparticles, and in particular our approach to this. You know, there's a lot of effort in making nanomaterials, and I think if you think about nanomaterials, there's basically two general approaches for the fabrication of nanomaterials. There's sort of the bottom-up self-assembly, atoms into clusters of atoms like quantum dots, or molecules into self-assembled supermolecular structures like micelles and liposomes. This has pretty much been dominated by the world of chemists of different types. And then there's this whole other class of nanomanufacturing that's more the domain of the semiconductor industry, these so-called top-down lithographic techniques, which have been used to drive the fabrication of different types of microelectronic devices and microelectromechanical type systems. And my group is interested in trying to tap these types of techniques for making organic nanoparticles useful in a variety of different biological systems. Eric Lander mentioned yesterday about Moore's Law, and it's interesting to think about Moore's Law in a biological context. Back in the early 70s, when folks were buying computers, the chip size was about the same as it is today, about a square centimeter, and one could cram about 2,000 transistors onto that chip because the minimum feature size of these transistors was about the size of a single cell, about 10 microns. 40 years later, you buy a computer, the chip is about the same size, but now the minimum feature size is on the order of the size of a virus particle. And you're able to achieve about multiple billions of these on the same relative area. And now we're talking about advanced precision technologies that are in the range for starting to probe biological systems. And this is a context that we want to apply. But obviously, these lithographic techniques are usually used for metals and metal oxides and are not conducive to biological systems. So my laboratory has pioneered a focus on translating these technologies into organic materials. And we developed a technique we call PRINT, particle replication and non-wetting templates, And basically what we do is we start with a standard silicon wafer, just like the standard lithographic techniques. We etch patterns into that wafer, and these are 100 nanometer features that are physically etched into the silicon wafer. We pour some different types of polymers on the surface of this wafer. This is a liquid fluoropolymer that has a positive spreading coefficient on just about all surfaces. It will wet every nook and cranny of this wafer at the nanoscale. It's designed to be photochemically cured in tens of seconds. So once it spreads and wets, we shine a light on it, and we peel off a membrane that now is basically a mold derived from that master template. But this mold is made out of a material that's got very similar characteristics to Teflon. It's a very low surface energy material. And what we typically do is take another material, another sheet or film, if you will, that has and I'm showing you here in red, a liquid that's going to be a precursor, a liquid or a polymer that's going to be a precursor to a therapeutic or a vaccine. And we basically spread that out on another sheet, and then we marry the two up, and we run these through a roller. And we do a film splitting technique, and we're able to uniquely fill these cavities without wetting the land area between the cavities. And now we have individual pools of liquids that we then solidify, and I use that term broadly. We convert that liquid to a particle, either through chemistry or a phase change. And now we have individual particles that when we then take that film, marry that up with another material, another film that has an adhesive on it, and this could be simply a water-soluble adhesive, run that through a roller, transfer all the particles out onto that film, and then we harvest those particles by simply running that film through another roller with a bead of water, typically, dissolves away that adhesive, have basically a dispersion of particles that we can lyophilize and have individual particles. So it's a templated manufacturing technique for making nanoparticles. And we've now developed this technique and pushed it all the way towards GMP clinical capability. This is a roll-to-roll machine at our company, Liquidia. We get thousands of linear feet of this film. We have a miniature version of this in our laboratory at the University of North Carolina. This is now GMP compliant, and it will support actually through phase 2 clinical trials that we're starting in vaccines, and I'll walk you through some of that. The particles that we fabricate now have the precision and uniformity of the microelectronics industry. You know, etching these features on a wafer, you can do it on this side of the wafer or this side of the wafer, and these techniques are highly reproducible. And so you end up with populations of extremely uniform particles. This is dynamic light scattering of our particles. So we're more often than not, this is a bell curve or sometimes even a mountain range of different maxima. We have, in fact, really monodispersed particles of controlled size and shape. And we've been able to fabricate a range of particles of different characteristic shapes. These are micron scale particles where one could vary aspect ratio. We're very interested in inhalation products. I'm not going to talk about that today, but we can control particles in a micron size range, which is really important for deposition in the airway. These even have nanoscale etched structures on the surface of microscopic particles to prevent contact. These particles here are regio-specifically labeled because when the particles are in the mold, there's one face exposed. We can conjugate antibodies and peptides to that one surface. We can control modulus and a number of things. These are the particles on the nanometer scale. So these are tiny little discs that are just over 100 nanometers in diameter and 35 nanometers thick. So keep in mind, a red blood cell would be about 8,000 nanometers in diameter. These are getting on the length scale of individual macromolecular chain dimensions. These are the size of many liposomal products, and we're very interested in controlling the aspect ratio and making filamentous-like particles of controlled size and even flexibility. As you see, as we lift them off the surface, you can see they're deformable. And we have master templates where we can keep one dimension constant, and we can systematically vary the other dimension. And we're very interested in how objects like this interface both at the cellular level as well as delivered systemically through different routes of administration. These are examples of some of the particles we use most extensively in our laboratory for parenteral delivery. These are 80 nanometer by 320 nanometer particles. These are the largest particles that we can fabricate that can be sterilized by filtration as we start thinking about that. So that's the bridge. If you want to go bigger, you have to work aseptically. but these are filterable and you can sterilize. And I'll show you the range of chemistry is extraordinary. We can make particles out of any chemistries, ranging from antibodies to polymers to pure drugs. And so what we have the ability then is systematically controlling for the first time key fundamental particle parameters of size and shape from the nanometer scale to the micron scale, and then cover a range of chemistries that allows us to probe surface chemistry in a very defined way, because the particles are initially in an unaggregated state, and we have regio-specific labeling capabilities. We can control chemical composition, and we can control modulus. And so with this toolbox, with basically what we consider nanometer-sized tools with precise dimensions, we're interested in probing different biological systems. So we start simply, and I'm going to go through a little bit of discussions first at the cellular level, and then I'll get to the systemic level. So this is a macrophage taking up particles of different size. And I showed an image here of a hex nut, obviously an unusual-looking object, but clearly a man-made particle. And we watched the different internalization processes as the particles enter the cell, the different endocytotic processes, macropinocytosis, Clathrin-mediated endocytosis, for example. We watch the internalization process. We use electron microscopy to watch where they're located in the cell, how they traffic within the cell as a function of size and shape. We measure the kinetics of uptake using flow cytometry and looking at that as a function of size and shape. And it's actually these particles here that are in our library of particles are the particles that are able to get internalized into cells, into non-phagocytic cells, the fastest over any other shape in our current library of particles. And they're not the smallest particles we have. And so there's something about the physics of this asymmetry, and it's actually very close in symmetry to many rod-like bacteria, and we're interested in the physics of that. And so that's beginning to understand some of the shape characteristics. We begin looking at how the particles are internalized and how they're trafficked. When you start having particles that have a fenestration or a hole in the center, what type of endosomal compartment does that initially form? How do those endosomes evolve into lysosomes, or what's unique? What's the pH environment within those endosomes as a function of time? And we begin looking at the details of these types of structures. We look as a function of time here, and this is a Clathrin-coated pit, and we can see typically the dogma was that the particles had to be smaller than the diameter of the pit in order to go internalized, but actually we see the remnants of Clathrin-coated pits on particles that are 200 nanometers. And we can start doing competition experiments with different types of inhibitors to start deducing the range of mechanisms by which cells internalize these particles. And so we're beginning to deduce that, and now we're interested in some of these intracellular trafficking as a function of size and shape. Obviously, size and shape is just one piece of the puzzle. The charge, or the zeta potential on the particles, plays an enormous role. What we find in non-phagocytic cells, particles that have a positive charge on them are attracted electrostatically to negatively charged cells, and you can easily get internalization of a wide range of particles, even up to microns in scale, that can get internalized into non-phagocytic cells. When you drop the zeta potential to the negative range, where everything else is constant, you can dramatically inhibit the uptake of particles. And so our strategy is, can we now, with this observation, keep particles with a negative charge and now override that negative charge with specific targeting ligands to do specific targeting and try to get the specificity associated with that? And this turned out to be a really powerful combination. We keep particles with a negative charge, and then we can start decorating them with different types of biological molecules or non-biological molecules on the surface to begin looking at targeting. And we try to do this in a careful way where we had different ligand classes. We would have a targeting ligand, and we would have controls that might be a peptide versus a scrambled peptide, or an antibody versus a isotype control. And I just want to show you some glimpse of this data. This is an example of transferrin targeting. As you know, transferrin receptor is overexpressed on a wide range of different cancer cells. This is an antibody, OKT9, that we conjugate onto the surface of these well-defined particles that have a negative charge. In HeLa cells, we have terrific uptake at very low particle concentrations. This is a multivalent particle with lots of these ligands on the surface. But relative to its isotype control, just IgG on the surface, we have less than 5% uptake. So we have really a huge window of specificity with this parameter of negatively charged particles with targeting ligands. This is 4-hour uptake and dosing, and then this is 72-hour tox. So this particle is not designed to have a cytotoxin in it. This was just a targeting study. And so we actually look at this as a sort of a plug-and-play approach that we could put different cargoes in with different targeting ligands and begin spelling these different approaches out. There's a range of different cancer cell lines that we've looked at that are all overexpress the transferrin receptor to different degrees. Shown in red here is the uptake versus the green, almost in a baseline here. And this almost tracks with the degree of overexpression of the transferrin receptor on a whole host of these different cancer cell lines. So we're beginning to start moving into various therapeutic approaches. This is Herceptin conjugated onto these particles. When I talk about 0% Herceptin, that's 100% IgG. So we're varying the amount of Herceptin antibody versus an isotype control and just varying the stoichiometry or ratio of these two. When you have no Herceptin, it's 100% IgG. You have the same story, very low uptake. And as you systematically increase the amount of Herceptin, you can get very good uptake. And this is a negative control cell line. It's negative for HER2 expression. So this is really effective at surface chemistry, and I just want to show you how we're using this very simple system and trying to move this into the clinic. So this is actually a commercial product that our company Liquidia is moving into the clinic in September for a very simple system. These are PLGA, lactic acid glycolic acid copolymer, particles that are these 80 by 320 nanometer particles that we can sterilize by filtration. We can combine them with just about any antigen, and we have a tremendous enhancement of the antigen associated with our particles. And so we're entering a clinical trial in the third quarter of this year, and we're moving into a variety of different other approaches. And it's a simple delivery system, and we're beginning to add different types of immunostimulatory molecules as cargoes to combine antigens with autostimulation. Other types of vaccines that we're working on are the so-called conjugate vaccines, which are combinations of polysaccharides and proteins. You think about pneumonia for a Prevnar. It's a $4 billion a year vaccine. That's a molecule that's designed by covalently attaching 13 different polysaccharides onto a protein, and it's designed the way an organic chemist would design them. And I'm an organic chemist, so I'll slam organic chemists. But it's a very complex synthesis to conjugate polysaccharides onto proteins. Using print, we can simply mix these polysaccharides at whatever ratio we want. We can cross-link the protein matrix. We're able to process these at just above room temperature. And we have tremendously effective vaccines where we can compare the performance versus Prevnar. These are the print-based conjugate systems. Each one of these points is an animal in a response. And you can see the print analogs are coming all out of the gate relative to Prevnar, and then we do a post-boost, and we have really great performance. So this is a powerful tool to begin systematically combining different types of molecules in a matrix. So that's all cellular-based approaches and some in vivo work with the vaccines intramuscular. What happens when we start varying size and shape and deformability for parenterally delivered particles? Where do they go? What role does size, shape, surface chemistry play? And we now have this toolbox to begin looking at that. We use different types of imaging beacons on the particles. This is copper 64, so we can do quantitative micropet CT imaging. And this is a fluorophore that we have on the particles. And I want to show you one story where we got some pretty well-developed data associated with this. And this is a story that's inspired by red blood cells and cancer cells. Red blood cells are very deformable when they're young. In humans, they're 8 microns in diameter, and mice are 6 microns. They're able to squeeze through 3-micron-sized pores in the sinusoids of the spleen. They can undergo 100% strain and squeeze through these different types of pores. When red blood cells get older, they get stiff, like many of us do when we get older, and they can't squeeze through these pores. And that's how the body will remove old red blood cells from young red blood cells. Subra Suresh here at MIT has been looking at the deformability of cells for many years. And he's wrote one of the most definitive papers out there. And what's known is that cancer cells, in fact, have an order of magnitude more deformability than their healthy cell analogs due to disruption of the cytoskeleton. And in fact, metastatic cancer cells are yet even more deformable than regular cancer cells. And this is a thought that people believe from a mechanobiology point of view is how these cells get into places they normally don't belong due to the deformability. So people have measured the deformability and modulus of these cells, and as a material scientist, we can systematically replicate modulus and begin looking at the effect in a rigorous way of the deformability and biodistribution. So here's an example of cells, red blood cell mimics that we make using print. This is a hydrogel made out of polyethylene glycol. We can vary the modulus by controlling the cross-link density. We put a red dye in there to guide your thinking that they actually look like red blood cells. And we think these are opportunities for actually loading them up with different biologics and different types of molecules that we want to deliver. This is a microfluidic channel, a 3-micron channel with 6-micron particles, because we're going to be working in mice. And you can see the particles going through these channels and actually deforming and squeezing through. In fact, we can do time-lapse photography, quantify the extent of extension and the amount of strain that these particles undergo. We can vary the gap and look at the viscoelastic recovery as they come through these channels and reform. When the particles are stiff, they get clogged, so we can vary the degree of deformability. We can begin doing in vivo experiments. So this is a tail vein injection of these particles. We use an intravital microscope sitting on the capillaries of the ear, and we can begin watching the flow of our particles through the capillaries. The story is, when the particles are soft, they're extremely well tolerated. These are 6-micron sized particles. When they're hard, the mice are highly distressed, there's pulmonary emboli, and we have to sack the animals. And so this is a very straightforward way to begin looking at deformability. We get hundreds of data points from the intravital microscopy, so we can fit the data very well. This is a 2-compartment fit, 350 data points, allows us to back out elimination half-lives and distribution half-lives as we vary the modulus here. And when we start getting into the deformability of a red blood cell, which is about 20 kilopascals, we start seeing a tremendous increase in our circulation times. We're starting getting elimination half-lives are getting into several days now. And they're quickly eliminated when they're stiff due to the pulmonary emboli. We can look at the lung tissue. This is when the particles are stiff. In red are our particles. We can see they're hung up in the lungs. And when they're soft, they go right through. And you can see the organ distribution changes dramatically. We start seeing the lung distribution at a certain modulus break, and then we start seeing accumulation in the spleen. And we see a persistent extended circulation in blood. But what's interesting is for these large particles, we see a very low liver uptake. And so we're interested in the implications and opportunities associated with particles that can do that. So a lot of that was done at 2 hours, and now we're going out to 5 days, and this is out to 5 days, and we see our particles staying in the spleen as a reservoir and continuing to circulate. They're not cleared by macrophages. They're sort of held up in the spleen. And we're now getting into these very long half-lives in these systems, and we're interested in these as long circulating particles. But these are very low modulus mesh-like particles that are very deformable. They're never going to hold a drug just through sequestration because there's too much mesh size there and they could leak out. So what we're beginning to do is thinking about ways of conjugating drugs. We're also interested in now moving these particles down to smaller sizes. And so here's now, out of the same degree of softness, these types of particles at 200 nanometers, 80 by 190 nanometers, these filamentous particles, and we're now systematically going through and trying to develop a biodistribution map as a function of size, shape, and modulus in these systems. Well, obviously, there's a lot of interest in being able to conjugate drugs and bringing drugs into these types of systems, whether they're biological or small molecule toxins. Obviously, I've shown you we've been using different types of imaging beacons for some time. And so we're interested in, with these very soft particles, that we're going to have to covalently attach the drug because the mesh size is too high. There's obviously a lot of different pro-drug strategies that people have used in the literature for linking a a drug to some drug delivery vehicle, whatever it might be. These all have some challenging issues. We, on the other hand, have developed a novel linking chemistry that is based on simply siloethers. Organic chemists have been using siloethers for many, many years as a protecting group. We like to use it as a pro-drug linking strategy. As one varies the substituents on a silicon atom, you can change the rates of hydrolysis over 7 orders of magnitude, from dimethyl to T-butyl, and use that way to control the release of the drug separate from the biodistribution of where the particle's going, and try to combine all these into a common strategy. And I'll just show you one example of a drug that we're doing this approach with, a campotecan-type drug with a siloether linker group, and we covalently attach this into these soft deformable particles. So this is the kinds of experiments that are ongoing now. When you can start linking drugs or trapping drugs into these particles, it can be like a Trojan horse if you're looking at an intracellular uptake mechanism. This is particles loaded up with drug, and we can design the chemistry such that the drug only comes off in the lower pH inside a cell versus the higher pH outside the cell. We can start using stimuli of internalization for deleting drugs. And this is a YouTube quality video here that we love to stare at. And there's a lot of detail here I'd love to walk you through at some point, but let me move on. When we think translationally, a lot of people for the FDA are going to want matrices that they're used to dealing with. And PLGA, Bob has been using PLGA for a long time. It's the same material as our bioabsorbable stent, same material as bioabsorbable sutures. Typically, to trap drugs in PLGA, there's the classic sort of coacervation or emulsification or micellization approaches for using these types of systems. What we typically do is we draw films of these types of polymers with different types of drugs if we want to use PLGA as a matrix. We have to work under conditions where this film is homogeneous, because if we have phase separation where we're making very small particles, we'll have a heterogeneous population of particles. But when we do this right, we can now make particles of extraordinary control. This is simply PLGA and docetaxel, and we have encapsulation efficiencies that are derived from the simple molding process that are very high relative to kinetically trapping the drug. These are all the same size, all the same shape, but they vary dramatically in drug content, from 40% drug to no drug. We keep the size constant, we keep the zeta potential constant. And it has dramatic effects on the ability for dosing. This is empty particles, and then as we increase the amount of drug, these particles get more and more potent, and we can increase the potency versus Taxotere by several orders of magnitude by getting the drug content up, and this is plotted against drug concentration. So we're getting to the point of single particle cell deletion using these types of approaches. We're beginning to now look at the biodistribution of what I would call as a hard particle, PLGA, versus the soft ones I just showed you. These are different sized particles. We're looking at biodistribution. This is percent recovered dose. This is a little hard to read. This is tumor. So we're in the order of 10 to 15% of these particles at 24 hours accumulating in tumor. We still have 30 to 35% of the particles in circulation at 24 hours. This is an SKOV3 xenograph system. So we're trying to combine all these systems and begin looking at different type of efficacy. We're looking at different types of tumors. Each of these tumors have different fenestration characteristics, sort of in a Rakesh-Jain-like approach, looking at, with precision tools, trying to classify tumors, different sizes, different shapes, different deformability, how we can penetrate these different types of systems. And now begin going, even with PLGA, to these different sizes and a whole host of different sizes with simply PLGA particles loaded up with these drugs, and this is the direction that this is all going. We're starting to do some of our very first treatment. This is an intratumoral injection model. These are some mice. This is controls. This is Taxotere, and this is one of our very first systems. And we're getting some performance. We've got a long way to go yet, but it's early days in this strategy. One of the drugs that obviously people are very interested in are nucleic acid systems. These are siRNA. We cast films of siRNA and PLGA, making these simple systems. We don't pre-complex or make polyplexes. We make these films of PLGA and siRNA. We then harvest those with different types of lipids, and I'm showing you some commercial lipids of DOTAP and DOPE. We can add excess of these, and then we basically wash off all the excess. So we're close to sort of a monolayer of lipid on these particles. And these become very effective transfection agents. These are luciferase-expressing HeLa cell lines. We're getting into sort of single-digit nanomolar EC50 values, delivering siRNA relative to our controls. And we think we can do this with a wide variety of systems. We think we can do this with a wide range of different types of particles, and we're beginning a range of in vivo experiments. Taking a page out of Bob Langer's approach, this was an intratumoral injection model, a luciferase-expressing tumor line. This is a control panel here on the left that's a naked siRNA. This is an irrelevant sequence, and then this is the experimental arm showing a protected luciferase system, and we see a 50% diminution in the bioluminescence. And so we're beginning to work through these systems in a variety of different approaches. So that's all systemic delivery. Let me end on one last topic. As somebody is trying to get drugs into solid tumors, this is a real challenge. This is a real challenge whether it's a particle or a small molecule. And as I told you, Bob and I were doing some interventional cardiology with the stent work. And so we have launched a program to start taking the tools of interventional cardiology, minimally invasive tools, as a way of delivering locally or focal delivery of drugs using an electric field. So this is ionophoretic delivery, mild electric current to assist the local delivery of different types of drugs. Tuveson's laboratory is going to speak later this afternoon. He had this seminal paper in Science this past summer showing that, in fact, pancreatic cancer is one of the most challenging. This is a poorly vascularized tumor. It's shrouded in what he referred to as a fortress. But through hedgehog signaling pathways, if you could eliminate that fortress, you find that these pancreatic cancer cells are very susceptible to gemcitabine. But it's a delivery issue of getting that drug to these systems. So we've embarked upon a multi-faceted approach to design minimally invasive devices using a variety of different approaches, intravascular, endoscopic, and laparoscopic approaches, designing new types of devices, working with large animals. Like for stents, you have to work in a porcine model for device safety, but obviously there are no cancer models in large animals, so we have to do combinations of small animal studies with large animal studies. And I just want to show you a quick glimpse of some new work. This is a surgically implanted device. This is a semi-permeable membrane that's ionic. We call this an originating electrode. This is a counter electrode. Drug flows in through a 4-lumen tube here. We circulate the drug out. We apply a current, and we basically use electric field to drive drugs into these poorly vascularized tumors. This is a canine model, and we apply current for 60 minutes, and we compare IV infusion. And we can have tremendous increases in the amount of drug that we load up into these different types of organs. A 12-fold increase in the amount of drug, gemcitabine, into the pancreas with current than without current. And this is how much gemcitabine gets into the pancreas when delivered by IV. And so we can have tremendous increases in the amount of drug that we deliver. And the key is, when we deliver drug locally, we find no drug systemically. We're below limits of detection. So it's a focal delivery of the drug and gives us huge opportunities with doing that. We've now extended this to xenograft tumor models. We've miniaturized our device. This is a 3-millimeter device. This is on a patient-derived xenograft tumor in a mouse, and we're getting the same sort of results. We have tremendous increase of, this is actually now in the tumor, of gemcitabine in the tumor. And then when we do the IP approach that the Tuveson laboratory demonstrated, we get no drug in the tumor. And so we're beginning to use these tools for delivery. And we think beyond pancreatic cancer, there's a whole bunch of different cancers that we begin exploring and delivering drugs, antibodies, and particles. So with that, let me end. I think the key is there's great opportunities for engineering and applied chemistry for delivering a lot of different types of approaches, whether it's small molecules to biologics. We think the tools from the microelectronic industry gives us the precision and uniformity that heretofore has not been available in the particle world, and we think that size, shape, and modules can bring new insights in different types of treatments. I'm fortunate to have a terrific group. This is a partner in crime, Mary Napier, who runs our laboratory, a lot of different collaborators, and a great group of students, and a postdoc here with me, Hassan, Wefta Hassan is with me, and she's done a lot of this work. Vision without resources is just a hallucination, and fortunate to have great support from NIH, our CCNE, and financial support from Liquidity and NSF. And with that, I thank you, and thank you for letting me participate in your program.